Objective: to study about human reproduction, the male reproductive system, structure of testes. The testes is covered by the tunica vaginalis. It is a two-layered pouch of serous membrane. These are the peritoneal layers. They enclose the coelomic space filled with coelomic fluid. This allows sliding movement of the testes. The testes is enclosed in a fibrous envelope called the tunica albuginea. The tunica albuginea extends inwards into the testes to form septa. The septa incompletely divide the testes into 200 to 250 lobules. Each testicular lobule contains 2 or 3 highly coiled seminiferous tubules. Each seminiferous tubule is internally lined by the germinal epithelium. This is cuboidal epithelium. The germinal epithelium consists of undifferentiated male germ cells called spermatogonia. The spermatogonia produce the sperms by the process known as spermatogenesis. Different stages of development of the sperms can be seen in the seminiferous tubules. Sertoli cells, or sustentacular cells, or nurse cells, are present between the germinal epithelial cells in the seminiferous tubule. The Sertoli cells provide nourishment to the developing sperms. The sperms lie with their heads embedded in the Sertoli cells, drawing nourishment from them. The Sertoli cells produce factors that bring about the process of spermiogenesis, that is, transformation of the spermatids into sperms. They produce anti-Müllerian factor, AMF, which inhibits the development of the ovaries. They produce androgen binding protein, ABP. They form blood testes barrier. The Sertoli cells also produce a protein hormone, inhibin. Inhibin inhibits the secretion of the follicle-stimulating hormone, or FSH, by the anterior lobe of the pituitary gland. This is a negative feedback effect. The regions of the testes outside the seminiferous tubules are known as the interlobular spaces. They contain connective tissue. Interstitial cells of Leydig, or Leydig cells, are present in these spaces. Leydig cells are endocrine cells. Under the stimulatory effect of the luteinizing hormone, or LH, the Leydig cells produce the male sex hormones, or androgens. Testosterone is the main androgen. Testosterone controls the development of the male secondary sexual characters and spermatogenesis. Immunologically competent cells like B cells, T cells, dendritic cells, neutrophils, macrophages, and mast cells are also present in the connective tissue of the testes. The seminiferous tubules join together at their apices to form 20 to 30 straight tubules called tubuli recti. The tubuli recti enter the mediastinum testes and anastomose to form a network of tubules called rete testes. The mediastinum testes is a thick mass of connective tissue at the posterior border of the testes. The rete testes gives off 15 to 20 ciliated tubules called vasa efferentia. The vasa efferentia emerge near the upper pole of the testes and enter the epididymis. Each testicular lobule contains 2 or 3 highly coiled seminiferous tubules. Each seminiferous tubule is internally lined by the germinal epithelium. This is cuboidal epithelium. Sertoli cells, or sustentacular cells, or nurse cells, are present between the germinal epithelial cells in the seminiferous tubule. Leydig cells are endocrine cells. Under the stimulatory effect of the luteinizing hormone, or LH, the Leydig cells produce the male sex hormones, or androgens.